Welcome, everyone. It's really great to see so many people here. I'm Xin Huyu. I'm the chair of RMIT Professorial Academy. This is academy consists of about 20 RMIT distinguished professors. We act as a think tank and advisor and as an advocate of RMIT values. So today is one of the activities we've been conducting every year, every year where we deliver a few distinguished lectures by our distinguished professors. So today I have a great pleasure to chair the distinguished lecture by Professor Suresh Bhargava. But I don't think he needs any introduction. Certainly the topic, I think probably the reason why so many people come is because of topic. It's about gold, right? I remember in the early days, this is legend had it. I remember somebody said that when they give you the reward in early days with company, they give you the gold nugget instead of cash. You see, so you still have it, right? So other people are using it. Yeah, so today Suresh is basically talk about more than just the gold we have in our mind, put in our pockets, that's the wealth. It's about how to use that to make a change of people's life to the environment. So please join me to welcome Suresh.
I never thought that after 30 years of serving RMIT, I have to enter an exam again. I thought today, definitely, and since morning, okay, Suresh, that's your exam today. And I'm saying, the question was there is not that the paper was disclosed to me. The paper was disclosed to me, well, that's your topic. But the answer was so widespread, and I was just struggling to concentrate and how to deliver these lectures, because this may be my last lecture. I'm not saying this is my lecture, I'm saying this may be my last lecture, because it took about 2 decades to come in front of you to give a lecture. I am giving a lecture all over the world, of course. I'm just coming direct from China to addressing an international health industry forums over there in Yantai. And where 17 FRS is in there. But delivering lecture in RMIT to a wider audience, it was rare. And it is a great opportunity. Thank you, Jing. Thank you very much that giving me this opportunity to talk to me about my own audience. So let me share with you a couple of things. Not the lecture. Lecture is not important. You can find Google's, you can find my research, no problems. One of the secret, we don't know how he did it. How he did it. 30 years are quite a long time. 30 years, best part of your life, is quite a long time. How did he did it? Did he do it right? Wrong? Did he act as a leader or like me and me? Sometimes it is very common in the academia, me and me. Right? Academics sit most of the time in their boxes. Getting out of the boxes is very difficult unless they see some advantage of going out. Very rarely. But time is changing. So let me share with you, first of all, my journey. My journey with you. So I'm starting from there. I have no hesitation to say, I was grown in a poor family in India. I never saw the light until I was 8 years old in my life. But I never give up. That's the only trick. I never give up. I go through all those challenges in the life. No, I don't go through. I grow through all those challenges in the life. I completed my every class, just only one class, first class first. I completed my undergraduate when I was only 16, postgraduate 18. I ended in Imperial College London in 19 years old. So I was a lecturer in a degree college over there. I did not give up. The only trick was, and that's why I'm just sharing with you my experience with you. When I went there also, came from a college where there was no research backgrounds at all, and I entered as a Commonwealth Academic Staff Scholars. I joined Sir Geoffrey Wilkinson and Nobel laureates, but I end up with Sir Abbey Abels with Exeter University. I moved from Imperial to Exeter, so my degree classifies there as a Exeter University degrees, Southwest England, beautiful place. is in there too. Because the difference between a big guns and this gun, which is not too short either, big, huge. Eddie Abels was my father because I lost my father when I was just 19 years old. He was like my father. And that was the great gift England gave it to me, because not only he taught me that how to do the research, but he also taught me how to carry on my legacy, Suresh. Remain a gentleman. He gave me a definitions. Leaders are those who won the heart of the peoples. They don't fight with them. Anyone in your life, if you find somebody is fighting with you, he is definitely not a leader. They won the people. They don't fight with them. There are certain things. Then I continue. Of course, when I joined in RMIT, I came to here. Stateway directly sent me, directly parcel. I finished my PhD in 2.5 years. In fact, I never applied to ANU. He parceled me to Martin Bennett, because Martin Bennett did the PhD with the same supervisor when I did. And Martin Bennett was the FRS there. Gold chemistry start from here. ANU I joined over there, School of Chemistry. In 3.5 years' time, they offered me a permanent job there. I was in RSCS. I have the opportunity to analyze the lunar samples when the Apollo 11 went there. Is in there too. There, my interest is developing to apply it, because now I'm doing applied research. Is there. I joined CSRO after that. 3 years CSRO, Division of Fuel Technology, where my interest moved to the catalysis area. Because a big giant group with BHP Billiton's working on the partial oxidation of ethane, translation of natural gas into the oil through methanol technologies. Then we moved, carry on. Then I realized that I'm missing something. What I was missing something? Teaching. Then RMIT was converting, translating into advanced college to the as a university. That is exactly the year when I joined the RMIT, 1990. My journey begins in this university in 1990. Today, I'm an antique piece, right? Antique piece, corners, is in there, but valuable one, I believe. And when I started my journey, I quickly realized the department is not a research-oriented for sure at that times. Today is different, very different, is in there too. But at that times, there was not a single FTIR. When I started working my research, I used to work up to 20 hours contact times in my timetables. Can you imagine? I do research in CSIRO because my friends, they always remember in this world, the network always pay you dividend. The network always works. My senior scientist, Carl Fogger, who was a scientist over here, he said, Suresh, whole Clatham CSIRO lab is yours. Come on here. What do you need? We will give it to you. He set up the facility, he set up the FTIR, he set up the infrastructure. Whatever you need, just keep working over here. I first produced over there, and I believe I was told I was the first ARC Linkage grant recipient of this university with Alcoa. Then I quickly realized one thing is very sure. So if you want to work with the industry, you don't ask the money to them. You don't propose what I know. But you ask their problems. What is your problems? How can I help you? Because I asked many times here and there. I was in ARC Discovery on those days. I was interviewed at Monash University. But Monash University, I still remember the John Ruston's remarks on my ARC Discovery application. If you are in Melbourne, I would have given you. If you are in ANU, I would give you, Suresh. No, not in RMIT. You don't have any facilities whatsoever. What the research project says is in there too. Because there was the interview times. We used to get an interview on the ARC Discovery is in there too. But that's all right. I said, no problem at all. No heartbreaking at all. But then it continues, and then continues with the Alcoa. Then I spent my 3 months over there just to study their problems in Kwinana. I was offered a job at that time, but my family said, no, we are not going Western Australia. We are going to Melbourne. But I developed a friend there, Stephen Grocott. And then he was working, Suresh, okay, come over here, give a lecture. Spend the time, 3 months. And then I start learning their problem. There is about 68 scientists working in R&D. Alcoa is the largest producer of the alumina and aluminum in the world. $6 billion export income of this country alone. Pittsburgh, American companies, refineries in Western Australia. They have Alcoa Bayer process have hundreds of thousands of problems all the times. That's why their R&D is working over there. I learned their problems. I worked there. I talked them in my last presentation over there. I explained to the ATR FTIR. This technology was just coming up in the picture. And then we explained to them that, okay, how it can solve your, probably address some of your problems. They say, all right, $25,000, go and get an honors scholarship. We start a honors programs over here. That FTIR, and they also, what else you want? I need FTIR. They'll say, take it over here. The latest model of FTIR, alcohol transfer to RMIT. Okay, here it is, Perkinelma. We started working over there, and that project, we started from there, contributed a lot. If you go to the Google and search on Rumbo technology of alcohol, which they use in refineries everywhere, in their bauxite minings, where 24,000 mines, samples are coming by robotic technologies done there. The fundamentals of that are discovered in the lab over there. Still that old equipment doing the 90,000 samples by drift technology is still there. Is still in the FTIR. Then we establish a FTIR facilities over here. First, vibrational spectroscopic facilities, X-ray diffractions. Dr. Kay Lathams, she joined as a postdoc to me. That's the leadership starts over there. For 2 years, then she continues her journeys and in there. But she came to join the CWO techniques. CWO techniques, what we invented to Alcoa, it has gone to their pilot plant trials to America, and then it stays there. So my continuous journey is there. So we bought the Alcoas there. Then we start working with the Olympic dams to the uraniums. And then to the BHP 2005 to 2012 is a ASX listed companies. And then I joined lecturer there, lecturer, senior lecturer, associate professor, professor in digital chemistry. Is all those things were there. And now what I am, as you know, is in there too. Then I got my first award in 2006. At that time, it was a great. I was classified, I was told by RNI, you are the highest grant recipient of this university. 2006, I got my first Vice Chancellor Research Excellence Award. Continues, journey continues. Olympic dams, there was a mining booms of this Australia. So Olympic dams, Megan Clark's, and they comes, we work together. We have a plenty of project. Money was floating like anything at my centers and the groups. There was no hardships at that times at all. Those days is gone. I know those days is gone, is in there too. And a lot of things too. Then I joined also in 2008. I became a dean. I was a chemistry head of the chemistry discipline there. I was asked to join as a dean of the applied science for 3 years over there. And after that, that was a tough job, a really tough job, because I was given a dirty task to clean the floor, removing 12 academics around those. I feel that was a dirty job. Then I asked the then Vice Chancellor, Margaret Gardner, that look, that's not something I want to do. No, Suresh. I was told it's a one-way process. When you join the clubs, it goes only this way. Okay, I'm putting in the college. Then I joined as a Deputy Pro Vice Chancellor Internationals, looking after the world. Then I focus on India and South Asia. That's the journey. But the research journey continues around the globe. Now become more and more internationals. As I'm including the India and including the work with the Indian centers, the President of India awarded me honoris causa in Rajasthan University, first doctorate of science. I got in DSc over there. Then I continues. Then I also got I was working on the interface of chemical engineering and chemistries. Trust me, I won so many awards of chemical engineering. Chemiga, they got sick of me. Engineer of Australia told me, Suresh, no more. Because every year you are coming. Actually, I was feeling shame as well. And good thing about was every award come with $5,000. Nothing wrong with that. I secure, at least I got $20,000 from them. Every year somebody nominating, and after that, that's the only way perhaps they can get at me, give him a chemical medal, because after that you're not entitled for any more awards of the chemical engineering. So Institute of Engineers, but this award is a highest honor of any chemical engineers. First time awarded in 45 years of the Institute of Engineers to a non-engineers, non-chemical engineers, to a chemist. They say, okay, you are a practical engineers, POP. Practical process over there. Again, guys, do not take that I'm boosting myself. No, I'm sharing with you what I am today, how my journey was. Very simple way in my own language. So don't feel any way that I'm telling you. What I'm telling you is the facts. Whether you know it today or whether you know by your means, that's your choice. But I'm telling you that is the hardest journey can make you if you have the attitude of can do. You can also be, and every strength of mind will become better than me. I tell you one thing as well very clearly. People came out, other people came out, many people spin off in this university from me. They all are doing very, very well. And they will probably beat me, for sure. And I love them. Nobody happier as a teacher if his own students beat him. Particularly in terms of the recognitions, in terms of the research output and everything as well. That makes me happy. And the corporate project with Rio Tinto, we still with company India. And my latest job is connecting that all the all the collaborations, all the Indian national laboratories to RMIT in one shot. That model, again, the attitude was there. Attitude was there that do the things differently, which makes the difference. I was watching in my jobs that every university is collaborating another university. So what? Monash collaborates with IIT Bombay's. Now, University of Queensland going to IIT Delhi. India is a very popular place. Usually do the university to university collaboration. I said, no, do something different. So India has a network of like CSRO 39 national laboratories covering from top of the India to bottom of the India. And that comes under the Prime Minister office under ACSR Academy. We collaborate with them. That means all those 39 national laboratories are accessible to RMIT, the best of the best. The PhD students goes on there is only 1% selections out of 600,000 students appear in written exams. They take only 600. RMIT take only 25 out of those 600 every year. It's best of the best. This model got the Premier of Victoria Award, most innovative models. We beat Monash, we beat Melbourne's, we beat every Victorian universities, and I received a very top accolades there. Now 7 countries in the world copying the same model. I came from China, and China's very high ranking told me, here is the 150 PhDs there, Suresh, if you want to duplicate this model. I can sign the agreement with the university anytime when they want to. I was amazed. When I present to them, I was really amazed. I said, no, that's it. We want this one. So the journey continues. What I'm telling you, well, that's the hard pathways. An important thing is there. If you follow and read the other people's books, it's a different matters. What is new in there? You don't know one thing. Probably you know everything's there. One thing there. 800 apps are downloaded in today's world per second. 1 second, 4.8 million apps in 1 month. Do you know that technology is speeding, racing with the thoughts of minds? This is a different world. What it means, the different strengths will come tomorrow. Academic platform is a unique capabilities where the brightest mind come every year. All we need is trained teachers, innovators. How we can translate these brilliant minds into the innovators for tomorrow. So we have to do things. It doesn't mean the fundamentals will change, the science will change. No, the package will change. And that's why you see in RNI, we have a 7 ECP, different concept. Same thing, same fundamentals. Because simple rule in the life, if you are not updated, you will be outdated. If you are not updated, you are outdated. So you have to learn those things. Okay, these are updated models which are coming because the customers which is coming tomorrow, the researchers which are coming tomorrow will be different. Keeping in mind that, okay, you can go to ARC, discovery, linkage, all those things are very competitive. Industries are the first priorities with the RMIT as well. I develop different models. Well, that's the models. I say, let us use the power of collective intelligence. Why not? I alone can't do it. Actually, this came out accident because when I joined RMIT, I said, look, Suresh, apply in a 4 disciplines, you will get 1 grant. Unfortunately, on those days, I got all the 4. The problem is there, I can't let them go. The 4 small grants continues the 4 areas, then it becomes 5 areas. And I surprised that if you do the things together, the pleasures multiply, the pain reduce. If you have 4 collaborators, 4 celebrate because they joint grants, joint inventions. When you have unsuccess, your pain reduced to 1/4. Everybody is okay, trying to say, okay, come on, next time. Isn't that true? So doing the things collectively, you cannot lose anything. And remember in this word as well, the power of one is no power at all. You cannot do things alone. You cannot do things if you really want to achieve the greatest outcomes in the fastest way. A good way, a speedy way, well, that's the way to go. But remember, I will tell about it in the future as well. Remember, doing the partnerships, doing the collaborations, it takes times. And the Guru mantra was, you have to develop a trust among the team members. If you don't have a trust, it can become a painful exercise as well. Let me continue. So that was there. I was using that too. The collective intelligence is the process by which a large group of individuals gather, share knowledge, data, skills, and the purpose of solving special issues. Well, then somebody asked me what is new about it. I do all the times. I have on my paper, I have 5 collaborators, one in England, one in Germany, one in there, one in physics, one in there. Well, that's the old style, mate. Because that work takes time, because the person sitting over there, he will take his own time to do your work. So the problems, the model I presented to you, I have electronic engineers sitting just outside my office. I have a chemical engineers. I have a biologist, right? I have a chemical engineers. I have a different type of electronic disciplines. I have a pharmacologist who are sitting just outside, ready to me every time. These people talk about 4 times a day. They meet them together. And that was the difference. Nothing new. I was listening to the speech of the Cochlear CEOs, and he was telling that is the difference. My pupils talk 4 times a day. Other company, they're sitting in their boxes, different. They talk, but their speed of doing the things together, talking together, developing the trust together, developing the team together is a different model. And that's, guys, I'm telling you, if you really want to proceed in your ways, fastest way. There are students over here who produce 18 papers, top referee journal articles from here. Fastest move, collaborative way in the same universities. I know that too as well. They keep moving on those ways as well. And you can do it. You can also the groups can also point it out, this is wrong, it's not going to work. They can also point out that there is a market over there of your product, if you were right collaborators. So your life, which can go through the process all the times, or you search in there and here, can become a lot easier. And most importantly, in the research, if you are doing, if there is no fun, don't do it. So to create a fun, it is very important that you do in such a way that you enjoy it. And that's the things. We combine the capabilities of the individual intelligence minds to produce some of the best work in scientific research. We are fostering the strength of the individuals to create the knowledge economy at the academic platforms. And that's why I advocate that in the world, if the new change will come, it has to come from universities. If you want to have a real, the economies are basically recessing all the time. There is a talk about it there. And the problem is there, the trust is not there. Industries, why they spend so much R&D monies over there, they can have a portion of that on the academic platforms where the experts are there, infrastructure there, and they can train the graduates, because end of the day, they require those graduates for tomorrow. Who is going to run their industries? A skilled graduate. Why they want to train their graduate if the graduate itself is produced by the collaborations? They know it. I have the graduate. I will take him. He's in there. He's the right candidates. So cost can be reduced. All those things can be done in there too. So I challenged to my groups last year, well, ChemEc is entering into a stage 2. Stage 2 means translation research. Publish the papers, but publish publications with applications. It doesn't matter whether they invent a product which is going to make millions tomorrow. Doesn't matter. The matter was there. I want to introduce an attitude in their minds. You can do it also. You can also feel the pain, that how much pain is required to translate your publication from papers into a real product going to the market. Because outside market is very tough. Unless your product tell the customers that yes, I can solve your problems in very cheapest way, he is not interested. Every paper I read nowadays, every paper introduction you read half a page, they are going to solve all the problems. Do they? Do they not? Other things. 99% I notice there that it is a polishing of the same piece of the puzzle. Somebody is doing spherical particles, somebody doing cubical particles, somebody doing the trigon particles, somebody is doing that too. They're doing differently. They're publishing the top papers, no problems. But what is the difference is going to make? Is it going to reduce the cost? Is it going to be efficient? Is it going to be do that? So doing the things differently is right, but make sure it makes the difference. That is important too. So, first things, First thing was important. I noticed, and it was not accidental. Alcoa called me as a consultant. Alcoa CEO declared by 2025, all the refineries will be the mercury free. We are calling all the consultants from the work, Suresh, come on to Perth, isn't there? I attended, there's about 20 peoples, of course there too, and they were saying, Suresh, we are going to handle the problems, here's the money. Here's the money, here's the ground, here's the things, whatever you want to do it, let's go for it, do it. Because there too. So, but mercury, why mercury? Then I realized that mercury is really a big problem. It's a neurotoxin. 60,000 babies are born alone every year in America alone with mercury-related disease. You don't die with mercury. But what happens? The mental retardedness, slowness of the babies. Pregnant women's sea fish, methyl mercury is a poison, isn't there too? And is a poison, sometimes the poor people doesn't affect it. The mercury is coming from coal thermal power stations. Mercury is coming from the refineries. China contribute about 54% of the global mercury. India, 14%, 15%. There is a nice documentary of BBC on India, how this poor fellow is collecting all this gold. Because gold have a special affinity with the mercury. So how they are doing with the mercury ways of collecting the gold from the rubbish is in there too. That without knowing out that burning those gold amalgams is going to hurt them. There's a big goal. So that was the problems. We go to there and all those sort of things. You can see how this work. And now you see there is no mercury thermometer nowadays. There is no mercury lamp. Where they're gone? See? Because there is a problems. Also, dentists are not refused to put the mercury amalgams. They don't. It's all ceramics. There is a lady dentist, Lisa Martins. She's a big campaigner of free mercury-free dentistry. She works with us as well. Social impact is in there. She asked whether we can make a handheld device to measure the mercury when she's removing the mercury from the mouth of the pupils. So they work in there too. First research came out because we have things too. We have electrochemists there too. These people say that, Suresh, building with bubbles, can we prepare the high surface area nanoparticles? And I remember that research we have done a lot. We published about 600 papers altogether, isn't there? But we are talking about just gold today. Just gold, nothing but the gold, isn't there too? And that this ceramics, this building bubbles is there. It is opening. When I was giving a lecture in University of Tokyo, one of the professors told me, Suresh, it could be a metallic zeolites start. But long way to go. How you can control the bubble size. Bubble size is directly proportional to the particle holes, dimension of the particles, a lot of things to do. But the application, slowly and slowly, you've seen the journeys. So what happens over there, when we want to create the bubble is in there, or the spherical porous copper in there, first of all, this copper was reported in there too. And then you can see that on the bubble size, when a simple thing, electrolysis, you take the beaker, the electrodes, acid solutions, and the hydrogen start bubbling. The moment the hydrogen start bubbling from cathode, the metal particle is going to there as well. Somewhere you control the kinetics. The metal particle can go on the spheres. So hydrogen bubbles can act as a template. The bottom of the things, you can see as the hydrogen particle is going up, you got the small particles, you got the big particles. In the difference between the zeolites and this one, they have a uniform particles. In this one, you don't have a uniform particles. You don't have a uniform size in there. You have a macro size, you have a micro size, you have a dendrimeric structures. Benefit and limitation comes together in this one. Benefits, any big size of the molecules can go through, react. Surface area, increase like anything, is in there too. It can become source activity, goes up, is in there too. And this way, the first examples, we prepare the bimetallic copper. First example of the copper honeycomb structure, chemical communication, came out from these labs. Is in there too. Anthony Mullins, he was a great electrochemist. He said, Suresh, we can do it. So the trick is there, how you control the parameters. The electron densities is very important. The time is very important. The concentration of the thing is very important. And in this way, we not only could prepare the nanoparticles of the different metals, we can also create the bimetallic surfaces, trimetallic surfaces. The bimetallic surfaces and their high surface area, high active surface, these surfaces can be applied to many applications. And then I'm going to come back soon. Stay tuned. This, at the moment, we are doing just a fundamental research. Publishing a paper, like anything, is in there. No problems with that too. I mean, they are PhDs, all those things. Academic output is great, no problems. Gold, porous gold. Here you can see a dendrimeric structures. If you look the inside there, SEM is a dendrimeric structure there too. You put 0.1 molar KuBr4, 1.5 S2O4, 2 ampere per centimeter in 30 seconds. In 30 seconds, less than a minute, you can get a porous gold. Isn't there too? Important thing is about it that whenever we want to use a catalyst, and the catalyst people are sitting over here, we have to rejuvenate the catalyst. Water process, what you have to do, you have to, first of all, fresh the catalyst. Heat it up with the hydrogen atmosphere, make it fresh, and then use it. Here, hydrogen itself is a template, it's a matrix. You prepare on site fresh. Hydrogen is template. And then you are getting a neutral, a fresh catalyst, and use it. Stay with there too. You can prepare, but trick is there, controlling the parameters. The moment you use the parameter different, here we go. Then you won't get it. So reproducibility, we did a lot of work in this area. And not only in the gold, first time we realized in the gold by doing this electrocyclic voltammetry over there, well, there are in gold 0, there are some gold plus is available. It's a gold oxidation. There is active sites on the gold. I wish I could have things, but you can see there is active sites of the gold as well. I presented this paper in Cardiff. Graham Hutchings, he's a great person of the catalyst in the Ferrari discussion, and there was a lot of discussion. There was a lot of unacceptance how it can happen. It happens. The gold surface now is very active in there. Anything which is gold for 3.2 level, you can decorate that. What it means, then I can decorate these dendrimeric particles with different particles. Silver, lead, or not lead, is a Pb. And then the silver I can do, lead, platinum I can do, nickel I can't do it because nickel falls over there. And then what I can do, I can create these decorative surfaces. With the gold is the base, but I put the platinum in there. And all those surfaces can act enormous. In fact, there too, you can deposit the copper gold, different. All those surfaces can act at different type of behaviors, different type of catalytic activities. You can enhance them, you can decrease them, you can use them. Whatever the way you want, those surfaces are there to apply for. So one group can just create those surfaces again and again. Some groups can see what application we can see, no drama. And they both can collaborate. That was the year. Then we go to the copper gold, bimetallic. Again, I'm talking about gold. Gold is expensive, right? The copper is cheap. Use the copper matrix. Use the 95% of the gold copper and use just the 5% of the gold. Decorate with the gold. And that's why we did 0, 5, 10, 20 millimeter. And without losing the structure, still we make the honeycomb high surface area structures. That is there. And you can see that this way you can prepare. This way. And we did the simple reaction, sodium borohydrides. And to convert these para-nitrophenols to para-aminophenols. And then para-aminophenol is a precursor for paracetamol, headache drugs. Headache paracetamol. So para-aminophol, acylation reactions, and you got paracetamol. But the real process is the reduction process. In this way, you can get honeycomb copper, 15 minutes. Here, 3 minutes, room temperature, 7 cycles. Room temperature reactions, sodium borohydrides, you can do a complete conversions to para-nitrophenols to para-aminophenols in 3 minutes. There's no drama there too. So what is showing up? It is not only showing up a nice result, but it is also showing up the surface is very active. It can be used for anything where the reduction is required. You can use it. And different application can occur. Always remember where we academics most of the time forget. We forget the application of one things which is done in the metallurgy area, developed in the metallurgy area. It has a fantastic application in biotechnology. But biotechnology doesn't go and through look that. And that's the problems where the innovation doesn't comes. Right? But if you talk to the electronic engineers, if you talk to the biologist, if you talk to the pharmacologist, he will always looking what is in there for me. What can I do? What can I add? And that is the why I'm advocating strongly. This is a power of collective intelligence used in a collaborative innovation ways. Isn't there too? And that works. Isn't there too? Again, we use this is search activities. You can see how the search is active. We can prove it, all those sort of things, all those papers were published, and the copper sulfate and gold there too. Search activities are the main things. If you can create a surface with a high surface surface enhanced Raman peaks, it can be a potential for the biosensors. Is in there too. So we did a lot of work in this area. I'm not going to show in there, but the one I'm showing that too, the surface which is showing to be plane is showing away quite a nice peaks. So they are active. They're showing up, yes, this surface can be exploited in there too. Then come to the stage 2. We start thinking, okay, electrolysis is going on. Can we do it? What we can do it? How we can create a permanent type of surfaces which can be used for different application. Here is electrolysis gold by gold. And what we did over there too, that gold by gold, we could create by different parameters, different concentrations, gold nanospikes. And we could do it not only on the surface, we can do on the QCM crystals. The moment we got the QCM crystals, and again is a power collaborations, electronic engineers say, can you do on the QCM crystal? Because QCM can act as a sensor. And that's exactly we did. But while we did, the science was brilliant. Somehow using a lead acetate is in there as a growth directing agents, we could stop all the faces of the gold. Only the gold goes gold 111. Mainly gold 111. And the gold 111, we found a properties which has a very strong properties to go to the mercury, but dissolve as well as the fast. Right? To act any surface, any material as a sense, you want to see, okay, when it is there, you can sense it. When it is not there, you should not sense it. So when the mercury is coming in the air, we should get the signals. But when mercury disappears from the air, the signal should disappear. Like a perfumes, somebody is passing by, yes, you can smell me, yes. This is it. But some person pass on there, no, I can't smell anything. Person is gone. You know that. And there's a lot of advertisement in perfumes, you can see that too. So that's the way, as you can see, these surfaces are there. Lead directing growth there too. We did a lot of chemistry in that. And without lead, with lead, surface area is there, surface is tactiles. And we can also optimize the whole process by increasing the lead directing agents, 0.5, so we can get the nice nano spikes. We can also get the OEOCl concentrations, enough gold spikes which can do a good job. Now we are doing engineerings, we are going to optimizations. I'm preparing a surface which can optimize, which can be used commercially, or which can be tested commercially for a while. Just not by 1 or 2 times, but several times, several weeks, several days. In fact, 6 months. Trial was done up to 6 months, is in there too. So we did that, is in there, as you can see. This is again, if you're controlling the Cl concentration, you can change the morphology. This time you can get the gold nanospikes. Gold nanospikes are the different surface area, is in there too. Different facet is in there, different application you can discover. All those sort of things. So that can be seen, is in there, and you can see still gold 11 is in there. And these can be doped on the survey as well. Now tell me about the sensing development. So what happens? This is a QCM. And that is the equation where the QCM is vibrate 1 megahertz, 10 megahertz, electronic engineers know, 100 megahertz. What it means, 10 million times per second it vibrates. And it is, by this equation, is mass sensitive, right? So any variation in the mass will tell you the variation in the frequency is in there too. So any deposit on that will tell me how much mercury in there. And that's exactly we did, is in there. So we can do that, if we can show them that how the mercury is going, and this peak is coming up. The frequency, delta F is coming up. So this measure is there, and it goes off, go back to 0. Sensor. So when the mercury is going in the flow gas, stop. Flow gas, no mercury, go back. This dissolve as well. Is in there. Perfect scenario. And then we not only use the plane surface, but we also use the nanospike surface there too. You can see the difference over there. The response is 200 times, 300, 198 times better. Is in there too. Is in there. So 180%, 900%. And not only that, we did try different concentration of the mercury in the air. 1%, 1 ppm, 2 ppm, 7 ppm. We increasing the concentration, the drop is going proportionally. No problem. The surface or the QCM is working perfectly as a sensor. Is in there. As you can see, up to the 10 ppm is in there. We can do a modified surface, non-modified surface. We tried now 89 degree. Why 89 degree? Because now I'm working with Alcoa. I'm working with the industries who's talking about how we can manage the mercury problem. He says, Suresh, the temperature on the refinery is 90 degree. So try to do it at 89 degree or 90 degree, because we want to see how close is practical. 89 degree is still there, 180% is there. This in like the surface, no problem. So in mercury, I mean, in short, I can tell you that what is going on, we are a lab, which is a unique lab in this country, in chemistry department over here, which can solve probably most of the mercury problem, a complete solution. CSRO worked there, ExxonMobil worked there, Newmont worked there, Agilent worked there, BHP worked there, Alcoa worked there. Any mercury problems, we are capable to do that. We have been told by publications, by anything, we are almost #1 in the world. Any paper published in the mercury comes to us, 99% chances it will come to reference. Isn't there too? This is the area which was a monopoly at the time. It is growing very fast because the law is becoming more and more harder every country. There was a no law about 10 years ago, so people don't worry about mercury, don't want to talk about it. But the law is getting now tough and tough. So people start talking. Even China, when there, people are talking every day. No, mercury is not acceptable. It can be done. We have to do it. We have to do it. Different problems, still the problems remain. Scientific problems are not solved over times. Still we are far away how we can detect the mercury in the plasma, blood. The Nature editor told me once, I was in Canada, he said, Suresh, okay, you did a wrong things because you published a paper somewhere else. Nature never published a paper which is known to anybody else, even media. You have to come first. Second, okay, do one thing, develop a sensing in the mercury in the blood, a sensing thing, and talk to me directly. I still have his number, but we haven't done that yet. It's still there too. So the translation results rule is there, very simple. That's what I'm trying to tell you. And now I'm going through again there too. How to get more from less. And that is the mantra of the business. How to get more from less. Now it is changing. How to get more from less for more. Because it's inclusive. I'm changing my views. I'm not doing any more scientific, environmental, or health research for money. No. I've decided. This is inclusive. It has to be passed on with everybody who needs it. Not for money at all. We will protect as much as we can do, but we will also share it if possible. Isn't there? So that is the mantra was there. And then stage 2, these guys, the champions are sitting somewhere over there. They accept these things and they develop this handheld device for checking the mercuries. Needs still a little bit more modifications, but can be done. But it will be done by engineers, electronic engineers, a physicist, a chemist, a chemical engineers, working probably 5 or 6 times a day. They only come to see me when they need my signature on a check or the orders. That's it. Is in there too. They talk and develop the plans, show the reports every time in there, and they accept the challenge. They show it. They show it to the chief scientist in my office that this is it. He was very happy that okay, and that is done by the students. That is done by the ECR. Again, I don't know whether this product will come in the market, and that has never been the objectives. The objective was to create an attitude in their mind, can do. Science is just not to publish a paper. Science is there to translate some things, to do some things, isn't there too? And that is the attitudes we want to change. RMIT want to change, I'm sure. All the ECP is designed for that, directed to that as well, is in that direction as well. So, remember one thing as well. If you want to test your product, do this test. This was our told there too. Your product is affordable. If you want to see how the market will take. If this product is scalable, if this product is sustainable, if this product is user-friendly, is your product is rapid, is your product is excellent, it is stand in the market, is your product is distinctive. It's the assured test. If you really want to see, if you're really greedy, if you really want to see that my product is great product, it will make money, well, test it. It will tell you whether it's worth it or not. It will tell you that, okay, whether you can sell in the market, because end of the day, it is the market who will tell you whether your product is sellable in the market or not. It doesn't matter you publish in Nature. It doesn't matter you publish in Science. It doesn't matter whatever you do. If your product, a product which is very cheap, very simple, is sellable in the market because it is only $2, quite user-friendly. No drama. Translation or the impact is different. A type of the impact where your own peers group cited your papers. H-index, fine. But who cite your papers? The same people who work in your area. That is the word? No. That is a very fine, tiny drop of the word. Well, that is a one impact, a impact, right? Which is very good for the excellence in knowledge, science, no problems. But the major impact should be seen by the peoples who pays the tax, who pays the bill of your research, isn't it? If you pay for something, you want to see what is for me in there. They want to see outside, when it will take times, how it's going to happen, that's things. Second thing is there, then we thought about it, metallo drugs. Second portion of there. So I thought when I talked to China is there, remediations, my first sentence with the forums was there, where it's about 17 top leaders were there. What is the use of the wealth if you don't have a health? And the health issues cannot be addressed just by developing a drug. Remediations is one thing. Prevention is also very important things. Prevent it before it happens to you. What is wrong with that? Why you wait for that? But if you can't stop it, well then, drug is necessary. Is there too, right? That is remediation. Previously, I'll show you the prevention. You can avoid the emissions in there. Well, that's it. We work on the cancer. And that research came out as a passion to me because I lost at least 2 of my close friends because of the cancer in the last 5 years. And then I say, well, it's a painful. It's not that they died with the cancer. I mean, many people died with the cancer every day. And who knows about us as well, about me as well? I don't know. But what was important there, the pain they go through using these chemotherapies, chemotherapies, and also going through this cisplatinum. So then we start thinking, these cisplatinums, I guarantee you, if we come to the market today, it will never go through FDA. But it was 20 years ago. Then there was nothing. It goes there. It is such a side effect, such a painful treatment, which gives you extra life for some things. It works there. And by the way, I was working in organometallic chemistry, gold compound for 3 decades. I start thinking that gold was used in Ayurvedic medicines for there too. It's a Midas touch. Is in there. 9.6 million deaths 2018, 150 by 2020, 19 estimate 136 people die every day just because of the cancer. It's a serious issue. And if this forum of these academic platforms of the research institutes or the research forums can add any value to it, worth it. That's why these people are paying you. Well, that's my view. Anyway. Council, as they say, that all those things are enormous, is in there too. So then what we did actually, I mean, this is again, 70% of the breast cancer cases, 80% of prostate cancer were diagnosed stage 1 and 2. Breast cancer, an alarming situation. I mean, these data are copied from websites anyway, from there. You can get it. But I'm showing to you in the context of my talk, there is a alarming data. It can happen to a young person, it can happen to an old person, it can happen to a woman, it can happen to a man, it can happen to a child, it can happen to anyone. It is issues, it can happen to a rich, it can happen to a poor. The only difference will be rich will probably get a good treatment, poor will not get a treatment, is over there too, that's the difference. But it is happening to the body, biological body, what we all have it. So we have a responsibility to answer it, okay? So what we did, that's the platinums. Though, first of all, let me explain to you, the platinum act in the body through DNA, is a DNA interactions. Why it acts, why it is effective. A lot of studies has been done because this drug is available in the market for several decades, is in there too. So it's done, it goes with DNAs and all those sort of things too. It is now the time has come, the many cancer cells are resistant to cisplatinum. They even does not affect by the cisplatinum at all. So there is a resistant cell as well in the cisplatinum as well. So there was a challenge. There was a challenge in there too. So what happens in there too? I working in the gold, and I know the gold chemistry very well. For 3 decades, I think I developed a reasonable knowledge. Not only that, I do have a masterpiece of the mines, the father of the gold chemistry in Munich, Germany, Professor Schmidt Bar. You see how many books he has written? How many? We call them in the father of gold chemistry. Martin Bennett, FRS from ANU, Research School of Chemistry. I'm working from the day I enter in this country. He is still with me. He is still as adjunct professor with me. Network pays, mate. Network is your net wealth. Trust me. Right? And once all these minds are there, and then all of a sudden I got these 2 brilliant minds, Srinivas Sudilka, pharmacologist and everything, collaboration with India's, and lately, very recently, Magdalena's over there. All the encouragement was there. Let us do it. Let us try it. It doesn't work. So what? We test it. So this is the one there too. So what theory I'm developing, and I'm sharing with you without protecting my IP, that gold can act as a carrier of a suitable drug. Selective because it can target the cancer cells. Gold in different forms can target. I know it can target because every tumor cell is different compositions. That's why they are known different one. As a prostate, as a cervicals, as a bone, as a lung, different. If we can target, if we can design some things, a metal ions, which can target that and attach this metal ions, attach that metal ions, a drug. It can go selectively there. Very simple. Isn't there? And that's exactly what we did. Isn't there too? But there is a history. That's what I was telling you. Gold has been used in China. Gold has been used in India. I took myself, my mother took my, that gold Soran Basam, eating that Soran Basam without any problems. Is there different disease, but it's a rich medicines over there. So a lot of people's, and also in Ayurvedic medicine there too, there is Europhanes, I will talk about it. But in the modern days, let me tell you that gold cyanide was claimed one time killing the TB. This man, Robert Koch, got the Nobel Prize at the times, discovering that. Later on, it found it not true. Lot of side effects and not probably true in there too. But those days, probably the in vivo studies was not that common. It's the start of the century when we got it. But it opened the gate. It opened the gate. It opened the minds. Yes, there is something there which can work. Is in there too. There's the market drugs which you can buy from medical stores right now. Is there too. Aeronoflins is in there too, which is used as a treatment of the rheumatoid arthritis. Is in there too. There's a drugs. Gold is available. Gold can be used without any questions. One of the scientists from University of Missouri told me up to 5 milligrams of gold is biologically acceptable in the kidney, Suresh. No drama is in there at all. So we know that some background there. And starting from the gold nanoparticles, I'm not going to test that, but there's a gold nanoparticles, ample of work has been done. Champions are in Melbourne unis, champions are Queensland universities, everywhere. Gold nanoparticles, carrying the drugs, carry several amount of the drugs, go there, do the jobs and everything. That's the one thing as well. It has an advantage, it has a disadvantage. No question at all. Isn't there too? But second thing what we did, attaching a biological molecules with a gold in one hand and take that gold to the target cell and then act. Is in there too, and that we start working. There's the first papers we published in the Florbeans Gold 1 complex over there, European Inorganic Chemistry. It goes to the hot paper of the year straightway in the journals in there too. We start entering now in different area, totally new area. 5 years ago, 4 years ago, absolutely new area for us in there too. And that's why I'm showing you the Gold 1 complexes. The one melanoma complex, you can see that cisplatinum is showing 23.5 and our complex is showing 1.53. Enormous increase, several fold, remarkable increase. Not only that, we tested. What we got in the team now, we not only got the Madeline over there, but we also got the Glenn, Dr. Glenn. He's MD, he's a real doctor. He's a CEO of the Biocurate, Melbourne and Monash University. He comes probably every month, giving a very tough task all the times to my pupils in there. Do that, do that, do that, do that. Because he's thinking, if this drug goes to the market, what will happen to the customer? What will happen to the patient? Is in there too. So we work in there too. Zebrafish, we found Donald's, is in the Bandura. We start working on that too, because that also give you simple things, learning to my boys. My boys do the zebrafish Gold 1 complex as well. Show them very selective nature of the complex. Very selective. Not killing any other things. Zebrafish is there too. And then we start moving the Gold 1 phosphine complexes. Again, out of those two complexes in the PTAs is in there too. And that's there. You can see the cervical, prostate, and bone. Brilliant. Only thing is there that you have a screen number of compounds. You make those. And these compounds are novel, by the way, never been prepared before in the world. It's a new compounds what we synthesized over there too. So that works fine. Patent is in there too. That works fine. And then we go to the not only vitro to go to the vivo over there too. Melbourne Uni is there too. And then you can see that too. There's a control there, vagal control there. Gold 1 compounds where you can see the tumor has been reduced quite drastically. And you can see as compared to cisplatinums where the tumor is still gone but not gone. So it shows you very clearly in animal studies it works. Is in there too. So these are the exciting moments when the team say, wow, we're getting through. We're getting through. So we wait for a long time. And I must tell you that I have a very, very enthusiastic team group leader in there as well who never want to give up at all. No, never. Isn't there too? So then we go to the Gold 3 complexes here too. There's a release there too. We are not the only one in the world working in the world. many groups are working in the world. But in Australia, we slowly, for surely, establishing our space in metallodrugs. Value to RMIT is in there too. And I will show you. So we start working the gold 3 complexes is in there. We have a brilliant gold chemist. Crystal structure is there, single crystal structure is there. They not only form them, they characterize 100% the structures. Once I know the structures, I can work on the mechanisms very quickly. How it will act and how it will not act. Both things. Where it fails, where it works. Because you know A to Z of the molecules completely. Is in there too. That's the there too. As you can see, the crystal structure has been done, is in there. And as you can see, again, some of these compounds, which are ionics, doing very well in cervical cancers and also the basically on the bone cancers. 2 of these where the blues is there, you can see they're working very well, is in there too. And they are worth, a cases for the worth exploring further, is in there. Then 2 of my students prepare a compound because there was a debate was going on in the literature that gold 1 is good, gold 3 is good, someone goes reduced to gold 1 to gold 0. What we do? We say, all right, we prepare a molecules where the gold exists in one single molecule in the 2 oxidation state. First molecule in the literature. There, isn't there too? The gold exists in the 2 different oxidation state, gold 1 and 3. So I challenged to them, okay, we got the molecules, then we got this molecules was insoluble, so there was a problems. We tested to the pharmaceutical college of the Monash universities, and they say compound is very insoluble. We can, nowadays, you can deliver to the insoluble compound as well, and probably Madeline, I know that too. But then we make the ionic compound as well, where the gold is there, which is soluble, but itself is more soluble. But these compounds, in doing the vitro cells in the IC50 things, you see they're remarkable results. Of course, RMIT took a patent on those as well. They're showing a remarkable results, 200 times better than cisplatinums is in there too, gold 1 and 3. It opens the new pathways again, bringing the 2 oxygen state, killing the 2s, both oxygen state working nicely together to kill that tumor cells or to probably to the point where it is, to kill them as well. So we're looking from all directions, all thinkings, and then working in that state too. This is gold 1,3s, the mechanisms, how it works over there. It is nicely explained. Is a 3D spherical there, is a 2B plates, the spherical there too. So we tested about 30 to 200 times potent than the cisplatinums on different cancer cells. HLV, all those sensors, less talks to the normal cell as well. So it is selective, not killing or disturbing to the normal cell. It is also selective and also effective. Is in there too. Not only destroying there too, because the mechanisms of the gold compound go through the enzymes. Enzymes, tumor enzymes, particularly, where the gold goes attached, is in there too. Previously, the idea was there is the gold is the test, deliver the drugs over there, release that and go there too. It can be wasted. But we prepare the compound where the gold cannot leave this ligand. We have a strong hand and where is the free hands. So free hands goes to gold carbon bonds. What we created in these molecules is quite strong. It does not go dissociate so easily as compared to the simple coordination compounds over there. So that is done. And then we prepare number of compounds. As you can see, gold 1 and 3 ionic complexes. Its structure is complete. Its structure is confirmed. Chemistry is done completely. Completely recognitions. The molecules, no contaminations in there too. And then we show the gold 1 and 3 ionic compounds over there, where you can see for the cervical cancer, it is quite effective as compared to the cisplatinum. Cisplatinum is always our fundamental base. And then you can see in the vivo activities as well over there, is in there, to confirming by the Melbourne Uni, where we did the mice studies over there too. Benefit in vitro cytotoxicity, complex 4 containing the AU2 counter as a demonstration superior in vitro. Cytotoxicity, selectivity 6 and 5-fold more selective cytotoxicity. Mechanism, the cell growth inhibition there too. In vivo anti-tumor activities there in up to 47% compared to 29% of cisplatinums. So double roughly isn't there. In vivo anti-inflammatory activities is there in addition to the anti-inflammatory. Gold has a dual advantage because gold is also tested in the world, used, still sold in the medical there as a rheumatoid arthritis. So anti-arthritis compound. So we thought, let us develop a compound which can do the 2 jobs. It can also solve your anti-arthritis problems, but it is also kill the cancer. Isn't there too? So it is working for the both ends. So that is also these papers in there too. And where we are at the moment. So Basic research, almost done. It's a continuous process. We're doing it. In vitro studies, yes, we can do it. In vitro, in vivo studies, yes. Some of them is a continuous process. We are here. Human studies, clinical trial, no, not. Because unfortunately, we're running out of the fundings. So let us, the future, next year will tell where we are. Where we close down, so we continues. But we are determined to continue, isn't it, Nada? So show you the one paper just published with the cover page in the European chemistry. It goes viral in 2 weeks' time. I don't know, RMIT put a news, the news putting there too. It is 174 million people's potential viewers. It is going in Daily Mails. We getting a phone calls from German TV. We getting the SBS. We getting the, tomorrow the ABC is coming. Is in there too. Is a known there too. Is a cover page of the European. It is on the online, so you can see in there too. It gives the values of 1.7 millions. RMIT media told me $1.7 million values to RMIT. It is viral. It goes all over the world. Is in there too. So what we lose? Nothing. The reputation of university is the whole things. In the international world, there is only one currency exists, mate, is the reputation, nothing else. Everybody wants to study Oxford and Cambridge, isn't it? Stanford, MIT, why? Chemistry is the same everywhere. It's just the reputation, branding. And reputation does not grow overnight. It takes a lot of efforts. Lot of efforts, people like Zeng, people like leaders and all those people isn't there. Where it grows, where the university grows, where the you guys will come, okay, that's the place we are going, isn't there? Okay, well, that's the molecular engineering groups. You see in 4 years, just in 4 years, we have 23 Q1 papers. We have an advisor, Glenn and Madeleine. Of course, our team is there, 2 pharmacologists, 2 chemists is in there. And we have a collaboration, of course, with India, IICTs, where the things work over there too. And all those sort of things. You know, we, these 4 or 5 peoples working in a collaborative manners, we're getting there. We're getting there. It's a research which comes with the passions. And remember one thing, anything you do with a passion, you will reach to the excellence. Otherwise, it's okay. If you translate your okay to the excellence, I'm feeling great, well, do anything with your passion. You will feel great, for sure. Well, that's the difference. Isn't there too? Many patients keep writing every day. This is the lady who said, please disclose my emails to everyone. She is from RMIT. She got the patient surface. No, Suresh, disclose it. She said, I'll be the human trials. If you want to, if your drug's ready, call me. I'll be the first one to go into it. So these kind of emails, these kind of messages motivate you, passionate you, worth it. Not publishing a paper alone. We do. Nothing wrong with publishing a paper now. What I'm saying to you, but these type of emails give you a different type of feelings. Absolutely different type of feelings. Isn't there too? And that's why I'm telling you that think about yourself. If you're researchers, think about it, what difference I'm going to make. I'm going to make, because that's your life. And that's the best part of your life. Isn't it? You will target that. Okay, can I do that? And that's the teams. These are MEG groups. You can see over there who are working collectively. Professor Ishwat Bar is also in there. He's writing a chemical reviews, Impact Vector 53 review article with me right now, published next year. He's in there. But Ramakrishna is there, ICT. His students are here. Our Lakshmi is here. She's working also anti-cancerous over there, doing a wonderful jobs, completing in there too. All those. Last year, the paper published with this group by Rasika got a John A. Bardi Medal, Institute of Engineers, the best paper up among 400 papers. So all this work is getting slowly but surely recognized. Okay, what is done so far in my career life? We have only 7 patents, 5 successfully gone to the industries. Project FTR-based robotic technologies is going to the bauxite quality. CWO techniques, I'll go over. ATR FTR based on the lines by process. Utilization based on the magnesium sulfate, going to BHP Billiton, and mercury sensings is in there too. And now we move to the different areas. It is my nature to move to the one area to another because I'm an interdisciplinary scientist. Plus and minus. The plus is there, you enjoy your work. The minus is there, the world may or may not recognize you because you are not in a single field. You will struggle because the mindset is still not there. Mindset of working of an interdisciplinary way is just not there. It's still not there. It's still not there. It's coming. It's coming. I'm saying, I'm clearly seeing it's there, but it's not there yet. Because when I was talking to someone else today, is there what the hell you are doing in the things? You are a resource man. You are a top 1% resource man, Suresh and Suresh. What are you doing in the anti-cancers in there? He said, look at it, what we did in there. Though anybody, anywhere can make a difference. Only if you have an attitude of can do. If you accepted a defeat before you start, you are a loser. Nothing else. Isn't that? Well, some of my students, I'm very proud of my students. Some of these students has gone everywhere. I have produced more than 50, 60. Isn't there? He gone to Oxford. He goes to the Stanford. Mohammed got to the VC fellowships. Isn't there? 2 students got to DECRA last year from the PhD bench as well. Different universities. They go to different universities. But there are some of these students in there too. They have gone to the India as well. Visiting over there, so in there too. Well, that's the program I'm developing. So that's how I want to show you the last programs, what I'm developing over there. I was trying to produce a PhDs, and I tested these models to everywhere. Everybody loves it. That was the model I presented to China. He said, Suresh, that's why I say 150 PhDs. In China, one thing I noticed, they don't waste the time. They don't discuss anything else. Once they like it, here it is. Can you do it? Can we do it? I said, let me talk to my university first. Let me do it. Don't hurry. I'm not an authority here. I cannot sign anything. But they say, Suresh, can you do it? 150, 100 to 150. Go ahead. I'll talk to authorities. If they are happy, they can do it. Is in there too. But that's the models we are developing. Okay. I dedicate probably, as I said to my last lectures, to my 50 PhDs who celebrated my 50 PhDs completion 2 years ago, 2017, is in there. Out of 50, about 6 are from Europe, 3 from Germany, who came over there. One, Roxanne is working here. She is here from Belgium. And Tasso is not here. I don't know whether he's here. He's from Greece originally. Probably he's in Australia now. He's in there too. But that's the things. So research, mentorship, and leaderships, that is CAMEC and beyond. That's my job. That's my passion. And again, I repeat, the leaders are the one who won the pupil. Do not fight with them. If anyone you find somebody is fighting with you, make sure he's not a leader. But it comes, trust is a key. Trust is key. Otherwise, every day you will see hundreds of emails. Out of hundreds, 80 will be nonsense emails. You can work without that. The world is moving differently, right? So most important things, if you want to move smoothly, less painful, less stressful, that is the word there too. Well, that's the programs. We did it. I don't know I can show it to you or not, but that's the programs. And then the common phrase I have, that if we work together, if I become we, a different scenario. Even the illness become wellness. Right? When you share when the pain is shared among the 4, it reduces the pain. Pain is more important, is there too. Right? So it's very, very important, guys, that you are the leaders for tomorrow's. Our time, particularly my time's coming to close soon, is in there. But I can tell you that remember one thing. If you work together, you can't lose anything. Is in there too. Well, I always follow this saint in India, who always says that life is a great chance. Aim for the highest, seek for the highest, and you will get the highest. So what he's telling me, it is you who defeat yourself. Nobody else defeat you. It is you who defeat yourself. Once you determine, because the problem is there, you may have a dream, you may have a good dreams, but what happens there, halfway through, somewhere, some blockage will come, and you will say, look, don't worry about it, I'm not going to do it. And that's the point where you lose. Look the Obama president of US history. Look at his history, how he was grown and all those things. Look at many people's history. You will find they have done it. But then somebody say, look, I can't be that. No, you can be one of them. They have the 2 eyes, 1 nose, 1 mouth, 2 ears. Similar body. No difference. Only thing, they never give up. The moment you give up, that's your loss. Isn't that true? Remember one thing, challenge will come, problems will come, issues will come, no matter who you are in this world, no matter wherever you are in this world, they will come. Don't you think the President of America don't have problems? Plenty of problems. So don't complain anything. It's okay. I have a problems only. Okay, that problem, that problem. Problems will come. No problem at all. It's up to you. You want to defeat yourself or you want to win. Remember, I'm getting old, but experience never gets old. Thank you.
Thank you very much. Thank you very much, Suresh, for an excellent, outstanding talk. You not just to simply talk about research, you talk about your life experience and talk about the whole spectrum from the from very early beginning to the end. But I got the feeling this is. I'm hoping you're not concluding your life, right? I think this is just the beginning, in particular some of life experience. So we have a few minutes for any questions. Anybody has any questions to Suresh? I have a question, just for a non-chemical person. Why you have to choose gold? Is that because it's so stable? I mean, you can choose silver. You can choose all these different metals.
Yeah.
Why you have to focus on
Two
gold?
reasons.
Yeah.
I think that I started working with the gold, so I know the chemistry of the gold very well. Secondly, is there, I know from the history that gold is a bioacceptable element.
Okay.
When I give my mother with the honey from my own hand, and she survives, she died at 89. A lot of people say sellable market in the drugs in the India is a not only thing. The only thing is not tested. Secondly, European is in the market as well. So that confirmed me. Okay, well that's the element. On top of that, it came through as well. Well, yes, you're right. I got a gold donation from a company as well when we were doing that. So we have a gold. We have in ourselves. We have plenty of gold. Is in there, so we can use it. Probably I will left a lot more for the next people as well. People use it.
Honey, okay. Any questions? Comment.
I think you should ask the questions because that will give the impression you did not understand what I said. Or you understand 100%, which is impossible. So ask any questions,
Yeah.
I mean, I'm a free person. Anything, chemistry, non-chemistry. Yes?
What is it that makes the gold-based molecules able to target cancer cells while avoiding regular
Okay. Okay. Most of the cancer cells contain an element known as sulfur. Sulfur, selenium, thiols, and thiol derivatives, thyroid reductase type of molecules. Gold has a particular affinity towards going there. Right? That's the thought. This mechanism is actually right. So those enzymes, particularly when go there too, gold will go. We know the gold will go there. The moment the gold will go there, it will interact. The moment it interacts, the moiety which is attached to the gold will act. So I still believe the gold is acting as a carrier. It's a carrier for these drugs, what we attach there. So the ligands, what we attach to the gold is important, one thing. That can be tailor design, molecular design. B, interaction of the gold. And I know the tricks. Which gold is interacting with one? Not going to tell you. But it can be done. A lot of work, a lot of research work. Yeah.
Any more questions?
I'd like a comment. One thing that excited me from the data that you haven't mentioned, Suresh, was basically the fact that the gold compound, at least in vitro, remains to be seen in vivo, but at least in vitro was able to target some of the cells, the cancer cells that were resistant
Yeah.
to platinum. And one of the biggest clinical problems that we have, pretty much, is the emergence of platinum resistance during cancer treatment. And what happens is that when a cancer becomes platinum resistant, basically they have very few options.
Very good.
And then that's when the mortality comes.
Oh yeah, yeah.
So I think when we were chatting and you were telling me about your research, and I asked you, do you have this data? When
So
you
how many stars you are going to give me? 2, 3, 4, 5?
No, when you told me, yes, you have this data and you showed me the data, I got very excited. So just to say, this is something you didn't mention and I thought it should be mentioned.
Yes, I think, thank you. I think that's important.
Okay. One more question before we have refreshment, and we can continue. Of course, we can continue to talk to Suresh afterwards. So if there's none, so please join me to thank again, Suresh, for excellent talk.
much.